to Jiří Matas, who will say his conclusion on the work, and then we'll have 20-minute presentation by the candidate, after which we have time for the reviews. The reviewers will summarize their recommendation letters and question the candidate. The whole defense should typically take about 2 hours. Sometimes it's faster. The purpose is not to examine in depth, but to convince the committee that the votes should be positive. So the defendant is, as I said, Milan Šulc, who completed his master's studies in 2014. He finished summa cum laude, majoring in computer vision here at Czech Technical University in Prague. He started his PhD the same year, 2014. We saw that he has a quite good publication record. I checked yesterday, there were 158 Google Scholar citations already. So there is a significant increase since the time of the submission. And he has an age index of 8, which also increased since. He is an awardee of several prizes. Most notably, he has won the first place at FGVCX Fungi and FGVCX Flowers recognition challenges. These were at CVPR 2018 workshop. And then also the Expert Life CLEF 2018 plant identification task. He gained an extensive research experience visiting various groups, as listed in the CV. So the members of the committee see the additional files in the Teams meeting under the Files tab. There are the reviews, the full CV, and the PhD thesis itself. So if you wish, you can check. Let me see if. So we still have 3 inattentive members of the committee who did not click on the vote link in the vote email. Should you have any problems finding that email, please let me know in the chat or just call my attention. Somehow you can raise your hand in Teams to call my attention, or just type into the chat. Okay, so the work has 3 reviewers who kindly present all of them. Dr. Tony Pridmore, who is a professor at University of Nottingham, where he is heading the computer vision lab. Welcome, Dr. Pridmore.
Good morning.
Dr. Gabriela Čurka, who is principal scientist at Naval Labs Europe, France, and she's a member of 3D Vision Group there. Welcome and thank you for joining us.
Thank you for inviting me, and hello everyone.
And Dr. Tomáš Suk, who is present in person here in the room. Maybe you can wave in the camera. He is a senior research fellow at Czech Academy of Sciences, where he is a member of Department of Image Processing. Yeah, I'm here in the corner, so you will hear my voice most of the time, but don't see much of me. I'm a member of the committee together with Dr. Elena Shikudova, who is associate professor at Charles University, where she's a member of computer graphics group. Dr. Josef Šivec, who is a senior researcher at INRIA, with Department of Computer Science at ENS Paris, and he's jointly at the Czech Institute of Informatics, Robotics and Cybernetics here in Prague. Dr. Ondřej Chum, who is present in the room, is an associate professor at our university, a member of Center for Mission Perception. Dr. Boris Flach, who is online, he's associate professor in our university, also a member of CMP and head of the machine learning group. And my name is Radim Shara. I am associate professor here, member of Center for Mission Perception. So we have, in total, we have 8 members in the committee, 8 voting members. You may see some additional names among the participants. Maria Duchoslavova is a administrative support from the Dean's Office, together with Kamila Gregorova. There are some guests, Dr. Jana Noskova, who is from the CMP group, and that's all I see at the moment. We have a few more people who asked to join us in the Teams meeting, but they will not be present during the non-public part. Any questions? So let me state formally that there were no independent comments from outside of the committee received. Therefore, all of us are present. Therefore, I see no obstacle to proceeding. I would like to start with the supervisor's statement about the candidate before he starts presenting the work. So, Jirko, could you please say a few words?
Okay, yes. Hello, everybody. Thank you for the nice introduction. I will not just formally read the statement, because part of it has already been mentioned by Radim, that the work of Milan has some nice impact, as witnessed by the citations. I will speak a little bit about the topic. When Milan started, we thought that plant recognition is very interesting because it's an example of fine-grained recognition, which means recognition with many classes. It's natural outdoor environment that until then we worked mainly on sort of indoors, artificial objects, man-made objects, and it looked like an interesting change. Milan had to survive in his thesis the, switch in computer vision from classical methods into deep learning, and he managed to do that. And what was difficult maybe in the thesis is to find some theoretically interesting thing which would be beyond just trying different network architectures for the problem at hand. And he managed to do that in looking at the influence of prior probabilities and the interpretation of CNN as something estimating a posterior probability. And it turns out that in plants and fungi recognition, you have these type of problems, as you can imagine, which kinds of mushrooms grow changes over time and over space, and it's quite an interesting problem that nothing is static there. So the idea that you have a training phase where you prepare everything and then just test forever is not will not work that well. The fact that he has done at the technical level, his work is excellent, which is shown by the superb results in various challenges, and this showed both interest of both Google and some and sort of a Danish research center which is interested in plants and fungi mainly. And in the end, we have now a running system where the result of Milan Šulc's method is running, and it's being used by probably not in the winter, but in the summer, hundreds of people per day. So the thesis in the end has both some theoretical contribution and clear application potential. And the last thing I would say is that Milan worked independently. He's the guy who chases me, not the one that has to be chased. He has his own ideas, and he's very good at carrying out experiments. So I think has shown all the desired, I would say, attributes of someone who will be a good scientist, and therefore I was recommending the thesis to be accepted for defense, and yeah, I recommended him for the degree. Thank you.
Okay, thank you very much, Izzy, for a nice introduction. All right, so we are ready for the presentation. You have 20 minutes for the work. You may stay sitting, maybe it's easier, it's better for the audience. And you shall see the pointer, the mouse, so I think we will be able to follow. So it's your floor now.
Okay, thank you. So, as the title suggests, the thesis deals with fine-grained visual categorization, namely of plant and fungi species. Many different species identification tasks can be formulated depending on the type of observation, whether it's an image of a specified plant organ or maybe any image of the plant or fungi specimen at all, depending on the acquisition conditions, where there is a controlled background and viewpoint and light, or rather in the wild scenario where there is occlusion and some clutter in the scene, other objects. Depending on the number of species and the granularity of the decision, we typically work with fine-grained classification, and we work with up to 10,000 species. The thesis deals with the different species recognition tasks, ranging from bark classification or classification of leaves on white background, up to the in the wild scenario with the difficulties mentioned. We formulate each of the considered tasks as a single label classification problem on a given set of classes. So, as mentioned by Professor Matas, we chose the species recognition as a difficult task because the observation depends on many factors, not only on the genotype of the specimen, but also on the age, on the season where the picture is taken, on the local environment where it was living, and many other conditions. The classes also typically have small interclass differences because some species are even related and look very much alike, and but they also have high intraclass variability following the factors mentioned. So let us start with the more constrained tasks, which relate more to the early type of my PhD, which is recognition of bark. In this case, assuming that the bark texture covers all the image area, and recognition of leaf scans or photos of leaf on a white background that can easily be segmented. While the first case is quite naturally a texture recognition problem, leaf recognition was until then usually approached via shape descriptors or local features. But we showed that the leaf texture is actually a very good discriminative information, and we also proposed to treat separately the texture of the interior of the leaf and the texture on the border, which may give us some hint on the microstructures on the border of the leaf. We proposed a texture recognition method called fast features invariant to rotation and scale of texture, which extends our previously published work on texture classification and its use in species recognition. And it's based on the so-called local binary patterns, where the intensity of each pixel is compared to its neighborhood, and then for each of the neighbors ends up in a binary decision 1 or 0, and thus forming a binary code for the neighborhood. So the image is then represented by histograms of these local binary patterns, and to achieve rotation invariant description, we use the so-called histogram Fourier features, where discrete Fourier transform is applied to sets of features such as the rotations that up to the rotation would be the same pattern as shown in the rows on the right. Such features are computed on several scales in a scale space, both to do multi-scale description and also to achieve some sort of scale invariance. The classification is then done using support vector machines with explicit feature maps approximating either the histogram intersection kernel or the chi-square kernel. The results of this method or of the texture recognition approach were nice on the constrained tasks. We achieved the best results on the experimented bar dataset and the best results on all 6 experimented leaf datasets, achieving 99 or more than 99% accuracy on 5 of them, including 99.5% accuracy on the Middle European youth dataset, which has more than 150 species. The method at the time of publication also had state-of-the-art results in texture recognition, achieving more than 99% accuracy on many standard texture recognition datasets, and achieving nice scores on the more difficult texture datasets. But nowadays it was later outperformed by convolutional neural network approaches which further improve the classification scores on most of the texture recognition datasets. But still, I would say that this method may be considered competitive because it's very fast. So maybe deployed on CPUs without the GPU, it is much faster. And when we compared the proposed method to other methods, and our method is based solely on the grayscale intensity, we also tried to look at the color information, and we found a significant color bias in several standard texture recognition datasets, and we tried combining the methods with color descriptors and proposed improvements to global color descriptors. Let us move to the more complex scenario of species identification in the wild, where we have unconstrained conditions of how the picture is taken, maybe in its natural environment. So for this more complex task, we chose a deep learning approach and used deep convolutional neural networks. Now, typically, to gain good results with deep learning, we need large-scale databases. Luckily, such large-scale databases were available from computer vision challenges that were organized during the time of my PhD, like the LiveClef challenges or the challenges organized with the FGVC workshop at CVPR. Working on these challenges, we noticed that prior shift is a very common phenomenon for these tasks, where the distribution of species changes from training data, which sometimes is downloaded from the web, from online encyclopedias and so on, and it is then different on the test data, which often were real test data varied by users of a species identification application and so on. So we looked at the problem of prior shift, originally motivated in setting it in the context of CNN classifiers, where the typical way to train a convolutional neural network is by minimizing the gross entropy loss on the training set, which in fact is a maximum likelihood estimation of the parameters so that the network estimates posterior probabilities. So, thanks to this, our proposed methods or the methods that we work with are not specific to convolutional neural networks, but apply to all probabilistic classifiers based or probabilistic classifiers outputting estimates of the posteriors. So, if we assume that the appearance model, the probability of an image given a class remains unchanged, and only the class prior probabilities change, which is the definition of the task of prior shift, then we can adjust the posterior estimates simply following bias formula. We see that the adjustment should be proportional to the ratio of the new priors and the old priors. Here we use the index E to denote the evaluation time priors, so maybe on a test set or on a validation set. So typically the new priors are unknown, right? But in some cases we may consider them known. For example, in the case where we know that the system shall be evaluated on a uniform class distribution, which was the case of these three data sets. One is iNaturalist, the second is the GBC fungi, and the third is WebVision. That's the data set with the same classes as the ImageNet 2012, but downloaded from the web. So on the top plots you can see the distribution of the training set in blue and the distribution of the validation set in orange. And on the bottom we can see how the accuracy evolves during training in blue, and how the accuracy improves just simply adjusting to this known change in prior probabilities. So that's about 4% in the case of iNaturalist and the GBC fungi in the end, and about 1.5% in the case of WebVision. However, typically we don't know the new priors, but having a whole set of test data at hand, which in some cases happens, we can try to estimate the new priors. Science and others proposed the following EM algorithm to maximize the likelihood of the observations, which iteratively adjusts the posterior predictions and estimates the new priors. Duplessis and Sugiyama have later shown that the EM algorithm may not always result in the unique optimal value, so we decided to also experiment with direct optimization of the maximum likelihood objective, which leads to some problems. So in the end, we can work with convex optimization framework and use projected gradient ascent. Projected because we need always to stay within the probabilistic simplex. We also proposed a maximum a posteriori estimation approach, assuming hyperprior on the categorical priors. Namely, we use the symmetric Dirichlet distribution parameterized with alpha greater than 1, so that it favors dense distributions, mainly with the intention to avoid the sparse estimates where there would be zero probabilities, zero priors for some species or for some classes, which would be bad in case such would appear in the end. And because also this distribution is slope concave when parameterized with alpha greater than 1, it is suitable for our optimization because we can just add the following gradient components and continue using projected gradient descent. So we, in order to experiment with different training set distributions, we have generated several artificial subsets of the CIFAR 100 dataset following different class distributions, and then tested it on the full CIFAR test set. And the comparison shows that in this case, the maximum posterior approach always improves the results upon the baseline, which is not that surprising because here the hyperprior is favoring dense distributions, and indeed the test set is a uniform distribution. Here are results on 3 more real data sets, PlantClef 2017, FGBCX fungi, and web vision. So on the top, you can see how the log likelihood evolves during the optimization. Here we can see that the EM algorithm actually converges well and faster than the projected gradient ascent, but even that is very fast. Regarding the accuracy, on the bottom, we can see that in all cases, the maximum a posteriori approach seems to be kind of more robust. Even in the case of web vision, where actually all the estimation methods lead to decrease in the accuracy, but the maximum a posteriori leads to the smallest decrease. On the other hand, in PlantClef, it doesn't add that much accuracy, but it still improves noticeably by about 2% accuracy. But here, this is a special case because the test set was actually fairly sparse. There was a much larger number of classes than test examples. So here, the hyperparameter was actually, say, leading in a wrong but stable way. Let's have a look at the LifeClef plant identification challenges, which deal with recognition of all types of plants, herbs, trees, ferns. We started in 2016, when the challenge was about recognition of 1,000 plant species. It also had metadata available on the type of observations or the type of view, and about the observation ID, which allows to link observations of the same specimen, which has several pictures. In 2017, the challenge got bigger with 10,000 plant species, and in order to increase the number of images available, except for a training set which was downloaded from the Encyclopedia of Life, there was also a noisy training set which was downloaded by Google and Bing web search. So it was about 1.5 million noisy images, which sometimes contained something different than expected. A very similar scenario has been 1 year later in the Expert LifeClef 2018 challenge, which actually had the same training data as the previous challenges, but a new test set, and it was organized with the intention to make a comparison against human experts. And then in 2019, the number of species remained 10,000, but it was a new selected set of species from data deficient regions. So the training data was, the number of training data was much lower, and to that extent that about 20% of the species had less than 10 images in the training set. Some had only 1 image in the training set. So this made the task more difficult. Then we also joined the FGVC competitions organized with the CPR 2018 workshop, FGVCX fungi, which was about recognition of nearly 1,400 fungi species, FGVCX flowers classification challenge, which was about 1,000 species of flowering plants, and iNaturalist, which was a challenge in the recognition of 8,000 different species from different kingdoms. So not only plants and fungi, but also animals. There were also bacteria and other kingdoms. So that was a really large-scale challenge. Our approach was usually based, or was always based, on fine-tuning state-of-the-art CNN classifiers from ImageNet checkpoints. So as for the CNN architectures, we didn't do much of changes. It always worked as kind of, unfortunately in these challenges, it kind of has to be approached if it's not limited by combining predictions from an ensemble of models. It also helped to combine predictions from images with the same observation ID, because as I mentioned, this was available. I will talk about it later. At the beginning, we didn't know that was allowed. Filtering noisy data was important in the case of the noisy data sets downloaded from the web. Image augmentation helped both during training and as test time augmentation. We also found that using running averages of the trained variables improved the results quite noticeably. And adjusting to the prior shift, adjusting the classifier predictions, which we proposed, showed to be very useful in the challenges. We also found max out in some cases to improve the result. Here, max out is a function that basically splits the output of the previous layer into several splits and then takes a piecewise maximum over these. So how we used it was added right directly before the classification layer. So we would add, before that we would add one more full connected layer and then max out. So in the end it would be a piecewise maximum of linear functions. So you could understand it basically as a piecewise linear approximation of some convex function as a maximum of them. What we tried and didn't work was first handling the types of view known from the metadata, handling them separately with specialized classifiers. In the end, it showed up that it was better for us to train universal classifiers. This may be related both to noise in the metadata and to the fact that some of the views may be related, and it's hard to distinguish from what is a picture of leaf, what is a picture of branch, what is a picture of the whole specimen maybe, and so on. We also experimented with the so-called bootstrapping objective for training on noisy labels, but that also didn't bring us any improvement and rather made the learning a little less stable. And for the results on the challenges, in the live class challenges, we were always among the top 3 scoring teams. In the first year, we didn't know that we can combine predictions in the test set based on the metadata of the observation ID. So we found this would actually add us our 8% of MAP. In 2017, we also ended up third. Later, we tried it with a correct prior shift adjustment that would have improved the results. In 2018, we were finally first, achieving 88% accuracy on the test set of 10,000 plus species, which I think is a very nice result. In 2019, we also achieved the best scores, but unfortunately our submission counts as a post-challenge submission because we had a bug in the results export script and only found that after the results were released. In the FGVC competitions, we placed first in the FGVCX Fungi Recognition Challenge and FGVCX Flowers Recognition Challenge. We also placed fourth in the iNaturalist Challenge. And let's have a look at the comparison against human experts. So in the thesis there are more experiments described where we joined the effort of comparing the accuracy of the machine learning systems against human experts in plant identification. So this is one of the more recent results from the 2018 challenge, where I would say that our submissions actually show human level expert performance, because our best model outperforms 5 of 8 human experts, in the identification of species from a picture of a plant. Now, of course, the human experts are typically used to take a more active recognition approach, observe the whole specimen, not only recognize it from one given picture, but in the scenario when it would be one picture, they were allowed to use all available literature, and these are the results. In 2019, these are the results on the Amazonian flora. So it looks quite differently, but again, except for the two experts which seem to be very good in Amazonian flora, our method then outperformed other three experts. So then, reaching the human expert accuracy, it even becomes questionable how exactly we can interpret the results, because the test sets are also labeled by human experts. So maybe we are slowly approaching the level of label noise. Probably not in this case, where this is the case of the rare flora in the data efficient countries. And then for the fungi recognition, after winning the challenge, we got into touch with the Danish Mycological Society, and they have a system for collecting fungi observations, and they have an expert system where the experts can vote in what species the observation belongs to, and they asked us if it were actually possible to use our system in their application, in their mobile app and on their web. So we made it work. We are providing the system for already like 1.5 years, and it helped them to increase the number of users and to increase the interest in submitting the biological data observations, which are then shared publicly through GBIF, this database that shares biological observations in open source. So we introduced the service in October 2019, and from October to December we have basically, or the numbers of submitted sightings with images have basically doubled. Now I was able to also add the more recent results from this autumn and winter, because I was a little bit afraid that the 2019 results might be impressed, or maybe also so high because they had a TV spot on the Danish news showing the up, but also in 2020 the numbers went even up again. So to summarize, in the early part of the PhD thesis, we used texture recognition approach for the more constrained tasks, achieving more than 99% accuracy on most lead data sets and on many standard texture recognition data sets. The method is computationally efficient, but nowadays outperformed by state-of-the-art convolutional neural networks. We also managed to improve our methods by adding global color descriptors and by improving the color descriptors that were existing at the time. For the in the wild scenario, we took a deep learning approach to species classification. The approach won several competitions in fine-grained recognition challenges. We compared to the accuracy of human experts and showed that the system reached a human expert level of accuracy for recognizing from images. And the system is now applied in the citizen science project called the Atlas of Danish Fungi, where it helps to increase the number of data and the involvement of users in the collection of biodiversity data, which is then used for other research purposes. And by the way, now, thanks to the initiative of our co-author Lukáš Picek, we will probably also start working with Czech mycologists and kind of broaden the application. We also show that in the cases where it's relevant, it's important to take into account the prior shift, and we compared estimation of the new priors using maximum likelihood and the proposed maximum a posteriori estimation, which increased reliability and accuracy in several cases. This topic is also still alive. I'm now working with a good master student, Tomáš Šipka. We have a CVPR submission on this topic, and we propose new methods for prior estimation. Unfortunately, I cannot share the technical details because it's also being considered for a patent application by Toyota, but it looks very promising. So, thank you for.
Okay, so thank you very much for the nice presentation. I guess it took a little bit longer than 20 minutes.
Okay, I'm
but
sorry.
that's, okay. It was nice, it was easy to follow, so I enjoyed it. Okay, we have time for the reviews. As I said before, I will call you one by one, asking you to summarize your report and then ask additional questions. The candidate will respond. He will share his screen all the time, so you can refer to the presentation if you need. Okay, so Dr. Pridmore, would you like to start, please?
Okay. So thank you again for a very good presentation. To summarise my report, I felt that this was a very interesting thesis in that it mixed general methods around the estimation of the priors and correction for changes between the test and training sets, and had some generic methods in there that were potentially applicable to quite a lot of other areas, which was nice to see. And alongside that, a very practical problem that's getting more and more important out there in the real world. So detecting different plant and fungi species is important for agriculture, which is where I do a lot of my work now, environmental monitoring and a variety of other places too. So it was nice to see the two things in the one thesis. The methods that you came up with were very thoroughly experimentally evaluated and very clearly stated in the thesis what you were doing. I think it was a challenge for you that, like so many other people, and Andy Shuri said at the beginning, that the computer vision landscape changed dramatically during the time of your thesis, but I think you handled that very well. And the texture analysis methods that you had in the early part of the thesis were state of the art at the time. I think to move on. The competition approach I felt was interesting. I'm used to seeing people obviously address competitions. You've done a lot of them, which was nice to see. You had impressive performance and raises some interesting questions, I think, about the role of these competitions in the development of new methods. But it was good to see such impressive performance on such a wide range of tasks and challenges, and with the human analysis as well, comparison with human analysis, your methods also did very well there. So I think you have a good, solid practical background into the work. I think you have a good, solid theoretical background to the work. The methods were evaluated very well and performed very well. I found the thesis quite easy to read. I have made a list of the small number of typos that I find, but you always find typos in a document that size, and it was a very small number, so that's good. And the publications coming out of it were very strong, and I think somebody already noticed your Google citations are above average for someone at this stage in their career. So I think, all in all, I think it was a very nice piece of work and a good presentation at the end. In terms of questions I would like to ask, I think one of the things for me that was, I feel like, a theme through it was you focus very much on the challenges. And I wondered what your general experience of that and opinion of that was, having come to the end of it, because it felt like in some places you were perhaps addressing the data sets that were presented to you by the challenges rather than perhaps the ones that you would have chosen to do. So I just wondered if you were designing a challenge now, what properties would it have? And in particular, you said at one point that, I think it was the iNaturalist challenge, your correction method came forth because the data set they provided was small. Does your method suggest particular properties that these challenge data sets should have?
Mm-hmm. Thank you. That's a very nice question. Just for others, I don't have a slide prepared for it because it was not expected or it was not written in the review, but I really like the question.
Oh, I'm sorry, I didn't. So I should say to listening, I'm an Englishman. I've never done a PhD viva in the Czech Republic before, so I'm asking questions.
But it's okay, it's okay. I just want the other members to understand whether it's not a slide for it. It's a very nice question because indeed the challenges, I think that. Okay, so maybe starting from a high level, I think that it's the very nice contribution of these challenges is that they provide that they usually provide some data set as is to the general public or to the research public to compare the current methods or to compare against each other, which makes it suitable to see what the current state of the art is without the shift in time. On the other hand, the disadvantage is that usually there's quite a limited time to do the experiment, so it is somehow limited in terms of what one can actually try in the context of one challenge. Especially now at the times of deep learning, where usually learning the classifier takes several days, and the number of GPUs is often the bottleneck in the experimental part. From like what I think would be nice properties of challenges to be discussed. What I kind of said about is that there are no rules about like the size or the numbers of the models, which means that even when we were joining and we knew that everybody uses ensembles of models, we are also submitting predictions from an ensemble of models then. But if the rules, for example, said that only one classifier with, say, a given maximum number of operators is allowed, it would be probably more about really focusing on the improvements of the architecture or of the objective, and less of the power of ensembles and combined models. And then for how the data sets should look like, I think it's really hard to ask because, of course, it depends very much on what is the objective of the challenge, right? So in the plant quest challenges, for example, the primary objective was to study plant recognition, but then since 2017, I would say that the secondary objective was actually how to make use of noisy data that is available at much larger scales than the officially or by the expert validated samples. So yeah, I guess that's really hard to answer in general, but.
Okay, thank you. So I apologise for asking the wrong sort of question. I have to say I haven't done this before. And skimming through my report, I didn't actually put any questions in the report because that's not what we do in the UK. Okay, so Thank you for that. I do think it's an interesting part of your thesis that you have looked at and performed well in more challenges than most other PhD theses or groups have done. Yes, and I think your insights into what those challenges are like and so on is an interesting thing. Okay. Okay. So I didn't put any more questions into my report, and I'm concerned that I might not do this in the right way. So I think for the moment, if I can pass on to the next reviewer, because I was very happy with the content and form of the thesis.
Okay, thank you.
Thank you.
There'll be more chance later. So the next, please, Dr. Churka, would you like to continue?
Yes, thank. So first of all, I would like to congratulate for your presentation, which was very concise but very dense and really presented in a very short time. Thesis in France, it's 45 minutes, and also we don't have prepared questions in advance, so you might have some questions more, some thought I have, so I did not prepare neither sent in advance questions for you. But yeah, so coming back to the thesis, it was very nice, well written and very clear to follow. And concerning the content, it was a very challenging task you targeted, and you really go from the beginning trying to go from really handcrafted towards deep, so you really follow the evolution of the field, and that was really nice. And tackling more and more harder problems, and indeed because of this diverse appearance, complex structure, especially when you are in the wild, the problem is very challenging, but also very relevant and interesting, because the solutions can be interesting not only both for specialists but for the everyday people. So yes, the thesis, for this the subject was very interesting, and the solutions also. And also I appreciated that indeed the thesis brings both theoretical contributions and the integration of the model to the service was also very appreciated. And I don't want to repeat everything what was said. I just wanted to mention concerning the challenges that I also participated in quite a lot of challenge, and I know that it takes enormous time and enormous effort. So I was really impressed by the amount of challenges you participated and so successfully. So also I would like to congratulate both for that. Coming to questions, if I might also ask some non-prepared questions in advance. So one of my question is that you tackled a lot this fine-grained classification, and what I would have liked to see, but of course, as I said, you are always constrained by the amount. It's more to go towards something like semi-supervised learning. So how you would see, especially with this prior estimation and so on, how you would see as a next step to use the unlabeled data to improve your models. Can you tell a bit on that, evaluate a few words?
Yes, so thank you for the question. Well, the prior estimation itself in our case is unsupervised, right? Because that's what we do on a set of images available from the new distribution that is unlabeled. But I assume that your question relates more to training the classifier as of feature embedding and so on.
To go a step further, using this for sort of curriculum learning kind of things.
Yes, so we didn't really explore much in this direction. At some point, I was reading a lot literature on self-learning and hoping to apply something like that to our deep net, but we never really got into that. So I don't have any specific answer in terms of say methodology or results. What is probably closest, what is in the thesis to your question would be the bootstrapping clause, which I hope could be used in this sense, which basically resembles the basic self-learning algorithms, so that it combines some label, if available, with the predictions of the current classifier, right? But unfortunately for us, it didn't work well in these scenarios. I assume it's quite difficult.
I assume, yeah. Also, I was just curious about, I mean, now what you would see the next steps if it was to further improve. I mean, there are some new techniques like, for example, attention mechanisms could be something interesting to focus on more clearly on some of the aspects, and in fine-grained categorization, there are a few papers coming out on the subject. What do you think about how you would go towards this direction, how you see in these problems? Can you tell a few words about?
Yes, so actually I think that attention itself is still very promising and has much to offer in computer vision, as shown also by the very recent results, for example, in object detection, right? So there is this new attention-based detector from Facebook research. I think it was published in October or November, maybe October probably. So that shows that actually for attention to detail, the proposed application of the attention modules as proposed in the all you need is attention papers originally focusing on text recognition can have large impact on computer vision. And I assume that in these cases where indeed the network needs to learn some specialized traits on the individual, say, maybe not even on the species level, but for some groups of classes, it needs to learn some specific features, then maybe such mechanisms may be a good way to process such specific features.
I mean, I was thinking also about focusing, for example, in the looking in in details. I mean, how to learn how to focus on some parts more. For example, on birds, it was on birds, focusing on the head or where to focus. I guess here also something like focusing on specific parts more than on others could be some issues to explore.
Yes, and then there is also actually on the birds data set, right? There are already these papers that try to focus on these specific parts, for example. So yeah, I think this is a good direction, and also maybe to the question of the say semi-supervised, I would maybe also add weakly supervised. Because often it might be the case where the specimen is observed in the picture, but it's not actually a major object in the picture, or it only covers a small part, and this actually relates probably to the iNaturalist challenges, where this happened especially for species from some taxonomical groups. And I know that I think I'm not sure if it was the winner or if it was the Google machine vision team who also had an unofficial submission because they were also an organizer. I know that one of them approached this actually by training a detector that first detected where the specimen or where the specimen of interest is, and then this appeared in the training data too. So then train on only the detected part of the image, and probably this is something that could be interestingly done in a weakly supervised manner.
Mm-hmm.
probably even with some mechanisms similar to attention, where one could use the fact that it can process, say, the global context of the image, and then focus only on the part that is relevant for the task.
Mhm. And my last question would be a bit challenging in the sense that, yeah, you at the beginning you also handled some of the very clean data sets and nice background and so on. So I was wondering if how would have been possible to adapt some models which were trained on very clean data sets to some wild data set. And you see in a sense of the domain adaptation or transfer learning to go beyond just sort of fine tuning. But what do you, I mean, if you can just have some ideas, or if you, it's more your opinion. And I mean, I worked a lot on the meditation, so I'm curious about for this kind of data sets, how would this be possible, or would be interesting to address such problems in this way?
Mm-hmm.
What do you think?
Well, so from a very high level, again, I would say that there might be two ways one could take. First is try to get the less constrained task towards the more constrained, like for example, make a segmentation method that would segment the leaf from the background and then proceed as before. But indeed, a better approach would probably be some transfer learning. This is hard to answer to you. So I mean, Gabriela Ciurka is an expert. The right answer would probably be I would look into the book of Gabriela Ciurka on transfer learning to choose the best method.
No, it's just a big. I was just wondering if it could have been an interesting path to take, or it's better to really have a good data set, or which would be the best way of tackling if you have a few data only at the beginning, and what path would be the most promising, the weakly supervised, the semi-supervised, or adaptation. So that was more, as I said, a bit of high-level challenging questions. You as an expert of fungi and the plant, what do you think?
Well, yeah, I'm actually not sure what would be the best approach, but I assume that pre-training on the so if I imagine the scenario where you would have a large amount of the constrained task data and a low amount of the in the wild data, which is actually not what we have, because usually the constrained data sets were much smaller than the current in the wild data sets. But if it were like this, I suppose first pre-training maybe on the data from the constraint task, and then trying to make to adapt the classifier by learning on data from the new domain, maybe a good baseline. It depends how low the number of images were also, and I can imagine so. Yeah, I mean, I can imagine a number of experiment approaches, but I'm not sure if they are worth presenting.
Okay, no, it was just a high-level question. So anyway, I will let others, because I guess many others have questions. So thanks again, and it was a very nice presentation, and the thesis also. So I congratulate you for all this work, and it was a huge amount of work, I can imagine. So congratulations.
Thank you.
Thank you very much. So we have time for the third reviewer, Tomáš Suk, who is here in the room. We'll try to show him, but I don't think we have the means. So you will hear him, but will not see him unless you follow the overview image, in which is visible on the right. So Tomáš, it's your place. It's you.
I will be brief. The theses are very nice. I appreciate the especially work with a priori probability of individual species, because it is complicated problematics. And if I have some comment at all, I would welcome more details about the parts when the author describes his work with TensorFlow software. It wasn't sufficient for me to reproduce the results. So my question is if this level of details is sufficient for the author to reproduce these results. And second question, the author works both with photographs taken on controlled conditions and also with photographs in the wild. My question is if that older databases of the photographs in controlled conditions, if it has sense to maintain them. It means to add new species and so on.
Mm-hmm. So thank you for both questions. So I have the slides prepared here again. Now, for the first question, I'm sorry to hear that you had trouble to reproduce the results. Maybe I can send you some more details on that. But in general, most of the major part of the approach we use is using or is by default implemented in TensorFlow Slim. Or this is actually tricky because there's now also TensorFlow Slim, which is a slim version of the framework. Whereas what I'm speaking is TensorFlow/model/research/slim on GitHub, which is this repository. As such, in that it's very easy to reproduce the methods, with the exception of some things that we added that were not implemented, which one is max out, and the second one is bootstrapping. So I also added the code with these methods to my webpage. Now, thanks to your feedback, I didn't realize that we actually never shared these. Especially the implementation for bootstrapping, I remember, was tricky because indeed in TensorFlow, well, not in the new TensorFlow, but in TensorFlow version 1, which we were using at the time, one had to use these special concepts like hooks to add a function before or to change to change some default implemented behavior. So now I extended the amount of code and models. Well, no, the models are always on the web page, but I also added the changes in code to be there. But in general, I think reproducibility in our field is a hard topic as is. So even here for the challenges, for example, the noisy web data for PlantClef cannot now be retrieved in the same manner as before because it was such a huge amount of data that it was only shared as a list of web links to individual images and to some like backup location which is not working anymore, and the original locations often have changed in the meantime. So now one wouldn't be able to get all the images as were available in 2017, and then other issues, including the implementation, depend much and change much in time with how the frameworks evolve and how they are dependent on drivers. So nowadays, I would say it is simple to just share requirements for your PIP or for Honda, but back then in 2016, 2017, I had to compile TensorFlow from source code because the packages that were available at that time were not compatible with our drivers or with some software on our servers. So that basically also decreases the chance to reproduce exactly in the same environment. Luckily, these models still perform similarly, but we cannot really speak about 100% reproducibility in this scenario.
It may be my fault because my experience with that slim models is
I try to put in the thesis like the major hyperparameters, right? Like say the learning rate, the decay, the optimizer, and so on. But indeed, in these frameworks, I kind of relied on the fact that it's based that the rest that is not mentioned is always based on the default implementation in this given framework. So that for reproducibility it is doable, but at the same time, putting everything in the thesis, it's hard to find where is the border, right? Because say going down to the level of the details in the frameworks or in the software used and so on might be too much for a thesis, I think. But yeah, anyway, in case you would like to reproduce some of the results, I know that we have already exchanged some methodology in the past, not before the thesis, and I would definitely be happy to send you some more, say, instructions, or maybe if you send me how, like in which part you struggled in the re-implementation. So okay, so this is for the first question, and then the second question. Indeed, I agree with the reviewer that the topic, that the field has changed so rapidly that what at the beginning of my PhD seemed like a super challenging task is now among the tasks that one would perceive as the simpler tasks. And again, maybe just for the context for other members or others who are listening, Tomáš Šulc is asking the questions about whether data sets in a limited or in the constrained conditions make sense anymore as an author of one of the biggest leaf data sets on white background. And I think it's hard to answer because it depends on what the underlying goals are or what the underlying research questions are. So indeed, we show that with deep learning, so we had 99.5% accuracy with the text recognition approach and 99.9% accuracy with the deep learning approach, which may seem like the task is resolved. On the other hand, it depends. So for somebody who is a nature lover and wants to identify a species in the forest and has a white piece of paper and takes an image, it's probably a perfect score. On the other hand, if this were to be applied, say, in pharmaceutical industry, then probably 0.1 error rate, if it were changing, say, a healing plant for a poisonous one, would be completely untrustable to be used in the industry. So maybe in these specific cases, one should really pay close attention even to the 0.1% errors. Another thing is whether to build such data set separately, since now there are these platforms like the Global Biodiversity Information Facility, which is a great online platform where everybody or where institutions can submit their biological data and share it with others. and probably it's a great way to also allow others to add maybe observations that are of your interest. And then I can imagine a few research questions that may still benefit from such data sets. So now, I mean, the result that I showed was using a deep convolutional neural network model that which if you download in your mobile phone, okay, now that you have large internal memories, but a few years back your internal memory would be full already. So maybe such, say, nowadays simpler tasks can be a good benchmark for more efficient models, for some small models, which are not so easy to maybe compare on the more difficult tasks.
One comment to that second item: if 99.9% isn't sufficient score, the way how to increase it is a bigger resolution. I was told that when botanists can see texture of narration of fifth order, it is 100%.
Fifth order means after 5 times it splits.
Yes.
Okay.
I don't know precisely how DPI it means, but
That's interesting. Because I assume that has to be already quite a very nice zoom on the leaf.
I am not sure if 1200 dpi is sufficient, but it's way how to increase it. For me, it is sufficient. The answers.
Okay, thank you, Tomasz. So I think we have we can have one more round of questions. This time, everybody, the reviewers, the committee members can ask questions. So I call for questions now. Okay, someone is raising. Elena, okay.
Okay, hello everyone.
Hello.
You just touched the field I wanted to ask about. Did you investigate the inputs that were not successful in the classification? Like, was there some pattern in the images that were difficult to classify? For example, in the first database in the 2016, there was an image with the red fruit which was so out of focus that I cannot imagine it was classified correctly. So did you also investigate in this topic? So
in the first part where we did the bark and leaf recognition, we noticed that so the leaves were either scanned for pictures on a white background, and we were segmenting it with a very simple thresholded segmentation method. And looking at the error images, we found that quite often it related to wrong segmentation, especially in cases where there was a light reflection on the leaf. So the light reflection would cause that it would be almost white and therefore would be segmented out. So although we basically required shape consistency of the segmentation, so filling holes, in case that this reflection was on the side of the leaf, this would cause the segmentation to fail, and then then there were such images among the wrongly classified ones. For the deep learning approach, I mean, we couldn't exactly look at this usually in for the results of the challenge for like the next year until usually the next year the test set was shown, but we did a few tests on our validation sets, but I wouldn't say that there were exactly clear patterns. Like, of course, sometimes there were images where it wasn't really clear, but what is actually the thing in the image, or because we usually took validation sets from our training data, sometimes the training data were actually of not such a high quality as the test data, and sometimes there were weird images, but yeah, I would say we didn't find anything that would have otherwise a clear interpretation. Among the test data, looking at the training data, we have noticed that sometimes there were noisy samples even in the examples from Encyclopedia of Life, and sometimes there were, say, different modalities, or there were like pictures, say, from a microscopic from microscopic imagery rather than looking on a camera picture. So I assume that these training images were sometimes making it harder. On the other hand, in some challenges, like in the case of fungi recognition, the microscope imagery was quite common for some types of fungi because it contained not only fungi like mushrooms that we find in the forest, but also the fungi as spores, say, and their microscopic images were quite a common appearance.
Okay, thank you very much for your answer.
Thank you.
Okay, thank you. Boris Flak is raising his hand. I will pin you so that the student will see you, okay? So if you please switch on your camera again, okay?
Which I did.
Hello.
Hi, Milan. Can you hear me reasonably well? Shall I change something in my settings?
I can hear you well.
Okay. Milan, as you perhaps expect, I'm going to ask you a question related to the prior shift.
Okay.
So the two methods you've been discussing, you discussed in your thesis, both require a large test set, right? A sufficiently large test set.
Sufficiently large test set, yes.
Exactly. And I want to go into a slightly different direction. So imagine you trained your classifier, this deep network, and now we are at inference time, or test time, if you wish, and you get a relatively small sample of examples which you have to classify, and clearly you couldn't apply one of your methods because the statistics is too small, right? But imagine that instead I give you, as an omnipotent, as somebody omnipotent, is giving you the true relative frequencies of the classes in that test set.
In the small test set.
In the small test set.
Okay.
So you get a test set with 100 examples and somebody gives you the true relative frequencies. And what would you do then? Let's discuss options, how to deal with such a situation.
Yeah, so indeed, one could think that having the relative frequencies on the okay, one more question. My goal is to correctly classify this small test set, or to correctly estimate the priors from the new domain and apply it in the new domain? I mean,
I understand. No, there is no new domain. The overall, the prior probabilities haven't changed, but due to the, I don't know, due to the randomness in the choice of the test set, the frequencies in the test set are not equal to the true prior class probability. However, somebody tells you those frequencies, and your task is just to classify in this small sample in an optimal way.
So, indeed, the part in the optimal way makes it much harder, right? So, if I were to quickly answer like a baseline which is not optimal, it would be okay. I can use the class frequencies as the new priors, but indeed that does not set. So basically, what I can use in the case that you are saying is set a constraint that the resulting number of classifications for the given species is exactly this number that somebody told me, right? Or somebody that's given me.
Exactly.
Which is an additional information.
Exactly, you're right so far. But now let's discuss this constraint optimization task. So what is your objective function and what is your constraint? Not the constraint is clear, I guess, but what is your objective function?
Okay, so now that's a good question.
Why?
No, how to formulate it this way? I didn't consider this task so far, so let me think. Yeah, so what I can do is.
Now, what is usually the so when you are making decision in the simple case, in the vanilla case, what are you optimizing when classifying, when making the decision based on those probabilities you get, based on the posterior probabilities? So you get the posterior class probabilities, you make a decision. What are you optimizing? It's trivial, right, isn't it?
The classification loss, right?
Exactly. And so now we have this empirical classification loss plus this constraint.
Yes, but the classification loss itself I cannot put in the objective, right? Because I do not know the correct classification. So indeed that would be the final loss. But in order to make the sorry.
No, wait. So suppose, sorry that I have to roll back to this trivial thing. Suppose you know suppose that the posterior class probabilities are the true ones. And suppose that your loss function, and suppose that the loss function for misclassification is fixed. No, say in the simplest case 0, 1 loss. It's trivial, but please tell me the right thing, so to say. How are you then deciding given the class probabilities? What are you minimizing?
I'm minimizing the risk, which in this case...
The risk.
Because, yes.
Exactly, and the risk you can compute, right? So now we are minimizing the risk, but with this additional constraint, as you said.
Yes.
And is it and do you think it's tractable? How would you formulate this task?
Well, I'll have to think about the constraint. So, okay, my constraint would have the form that the number of decisions which is actually, okay, I could maybe make a, so I'm thinking about how to correctly represent the decisions, because putting something like argmax is probably not a good idea, so probably making like zeros and ones decisions may be a good idea to represent it, but now from the top of my head, it's not so easy to imagine for me now.
Yeah, maybe let us stop here. So I think now you are on the right track, and maybe for solving it, in the end you need a sheet of paper and a pen, but at least now you are on the right track. Thank you.
Thank you. And actually, it's a good question, because in some cases you might consider this scenario maybe for the future. It's like a good problem to look at, if you know the numbers are first.
Okay, thanks.
Thank you.
Okay, thank you, Boris. Eugene, you have a question?
Quick one. Thank you very much for the presentation. I just have a quick question. In some of the images you were showing, there is like an image of the fungi, of the mushroom from the bottom, for example. So I imagine of course I'm not an expert, but imagine if an expert looks at it.
We lost you.
We lost the sound. Yeah.
Can you hear me now?
Yes.
Yes.
Okay, oh, excellent. I see exactly. So imagine that. Okay, I'm not an expert, but imagine an expert is looking at the specimen from different viewpoints to examine this. In your framework, I imagine you did individual images, but would some kind of a multi-image setup help in this, or is it considered like can you submit multiple different images of the same species to get a better result? Do you have any experience with this? Do you see what I mean by this question?
Yes. So we have an experience with this from the challenges, where in some of them, namely in the plant life challenges, the information about there were numbers or sets of images for one specimen, and there was meta information for that, and we were using that. We didn't do anything too smart. We only combined them by averaging the predictions for the given picture. Now, for what you are mentioning, I think that the ideal would actually be an active recognition approach, but it's really hard and would probably require a lot of human expert knowledge in designing the system. In case the system would be able to tell you, I'm not sure between these 2 or 3 species, but please look maybe from the bottom of the head of the fungi or something like that. But we didn't touch any active recognition so far. But I think
thank you. It's interesting because imagine you have like a video signal that you may get to. That's an interesting thing. But I think it's fine. Thank you very much.
Thank you.
Thank you. Might be difficult to design a test data set for such scenarios.
Yes, indeed.
Actually, had a similar question, a simpler one. Do you refuse, reject data if it's defocused? For instance, we saw one of these examples mentioned, and perhaps others. That would be like a first step towards interactivity.
So in the system for microimages, currently there is no prior filtering, although we are considering that, mainly because then it complicates. So the system is based on human validation. So our machine learning system only makes a proposal of the species for the user to see immediately, to get an immediate answer. But then the observations get into the system, where a lot of human experts is connected, and then they manually label whether they think or do not think that the food or the mushroom belongs to a given species. And indeed, since the app is getting popular, there is also the issue that people are submitting random images just to try out whether the app sends an image or something like that. So we are also considering adding an extra, say, mushroom, non-mushroom classifier. And then for challenges, we did filter out the noisy data. in the way that, so in several ways in the different years, sometimes by first training on the more trusted data and then looking at the consensus of the noisy labels with the classifier prediction. So for example, if the labeled species among the top few predictions, it is considered that it's likely it's labeled correctly and otherwise not. Or we trained a special plant, not plant classifier for plant cleft 2019, which was used on the training data, and then the filter training data was used for training.
Okay, thank you. Any more questions from the reviewers or the rest of the committee? Actually, I have I don't know if it's a question, maybe it's just a topic for discussion that could be done offline. But, I'm amateur naturalist kind of.
Mm-hmm.
Maybe I know hundreds of plants and dozens of fungi, okay? So not very advanced.
Very nice.
So I often carry about a heavy book to help me identify species when I find something. Now, my experience is this. The first time I see something new, I don't know what is it. I try to find out. It's a lot of work because the standard way is to look at very detailed properties. Sometimes you have to wait for the rest of the year to see the seeds and things. Fine. Then I see it. Then I'm pretty sure it's what it is, because sometimes you are not sure based on the keys that help you identify. So in case I'm sure, which is sometimes happens, next time I see it, I immediately recognize it from properties that are different than in the book.
Mm-hmm.
I simply see it and it's clear to me, especially with mushrooms. They are very diverse, but first time I learn them, it's very difficult, but next time I see them, it's almost trivial to distinguish it from something else. Could you comment on this? Is this effect that some kind of systematic effect?
I don't know, but actually what came to my mind when you were saying this is actually that since you have to look in the books, look up the species, in case of like considered training loss for you, this is quite a high loss. And I'm actually, okay, this is more of like a critical comments for what we wrote in the thesis, but maybe if you were using an app, you would actually not remember it for the next time, because I think that the experience that many people have, for example, is GPS navigation, right? When I go to a new town without GPS navigation, then next time I go there, I know much better where to go than when I use GPS all the time, and it's so simple for me that my mind doesn't really pay the attention to remember, say, the problem I was solving. So I can imagine that if you spend, say, I don't know how much time you spend identifying new species, but probably like 10 minutes at least, right?
Much more.
Much more, right? So for plant identification, I remember that in the study of Sablatnik and Field, they claimed that in places with high biodiversity, experts can spend hours or days identifying one specimen. Then I assume that you kind of put so much focus in the observation that it writes into your mind, even with the parts that you were not really looking for in the book. But
it's a hypothesis. When I was young, if someone showed me a new species, I remembered it. And so the initial learning was like in terms of seconds, and then it lasted. So it's maybe a wonderful story. Anyway, thank you.
thank you.
So I would like to call for more questions, if any. And if not, we would close the session and proceed with internal discussion. So let me know if someone still needs discussion. So if not, it seems I can't. Yeah, I'm just answering to the supervisor's comment if he's supposed to participate in the closed session. The answer is yes. We may need to ask him something. Let me close the public part. We'll have interruption in the YouTube transmission, and we'll come back later after we discuss and decide on the result. Ne, nechte to. Okay, so finally the committee discussed and had a short summary of the presentation and question and answers. All of us were happy with what we heard. And then we voted in a secret vote. There were 8 voting members in a vote. And my great pleasure is to announce that all 8 votes were positive. So let me congratulate you. This day is a great day. And you will receive the title of PhD. This is not yet official. You will receive official papers from the Dean's office. Should you need faster outputs from them, we would have to help them to provide you with certification that you have passed later in the diploma. So again, thank you very much.
I thank you very much.
I should also thank all the committee, especially the reviewers, for their great work. Thank you. I enjoyed this defense. So we still are online. If you would like to say something, please do.
Congratulations.
Yeah, of course. Well done.
Yeah,
Thank you.
good job. Congratulations.
All right, we will stop now. We will stop the transmission to YouTube, and I will leave the session open in case you want to say something or discuss unofficially